Transcription is the process of getting information out of DNA and into RNA. It starts by having the DNA accessible. So euchromatin is I think like a lightly packed version of the chromatin. So that's going to be just physically accessible to proteins and other transcription factors and the whole transcription regulation process. So you can see the difference between euchromatin and heterochromatin is just how densely the DNA is kind of packed in the chromatin using various proteins. So you can see here 92% of the human genome is euchromatin. So there's only a small amount of it that's not accessible. And then it kind of all gets packed together during mitosis and meiosis when it's being duplicated. So the whole process of transcription is regulated. So it's in various organisms, it's a different situation. So bacteria has its own kind of regulation, and eukaryotes a slightly different kind of regulation. So mostly, I think you think of transcription factors as being the transcription regulation material. So here's a eukaryote showing enhancers working with promoters to regulate the transcription of DNA into RNA. So transcription has a few different steps involved, but basically, the transcription regulation kind of network has to bind the transcription factors and the various proteins to the promoters and the enhancers and kind of get that whole complex going. Then the DNA has to be unzipped to access one strand of DNA, and then RNA is generated, and then the RNA is processed or forms some structure on its own, or is transported out of the nucleus. So here you can see kind of this cartoon drawing of it, just the promoter again being next to the gene, the enhancer kind of being further away from the gene and the promoter, and then this kind of protein complex here needed to start the transcription of that gene. So a gene is essentially just something that creates a gene product. I think in the old days, a gene was something that made a protein, but now it makes some kind of RNA, which can then lead to some kind of polypeptide, which can lead to a protein. I'm looking for the list of RNA types. I had it sometime before. I guess just off the top of my head, there's obviously messenger RNA, transfer RNA, ribosomal RNA, microRNA, and various other small RNAs that are used for other post-transcriptional regulation. So once the transcription has been carried out, there's still other processes that can control if that RNA is used as it was intended, or if it's destroyed. So for like messenger RNA, there's regulation of it, but then there's also the processing of it. So it's modified as has a 5' cap and a polyadenylated tail. And then if there's a eukaryote that needs some RNA splicing to remove the introns, that's also carried out. And then the processed RNA, in this case the processed messenger RNA, is transported out of the nucleus to do its job of creating proteins in the cytoplasm. And just another kind of example, here's telomerase RNA. So the telomeres are the sections of DNA at the ends of the linear chromosome in eukaryotes. And they're just like a repeated pattern that during DNA duplication gets degraded. So there's a process of adding that repeated pattern back onto the ends of the chromosomes, and that protein needs a RNA template, in some ways similar to CRISPR, where you have a RNA kind of guiding a protein into what it should be doing. So here's another example of a gene that creates some RNA, and that RNA is a kind of in some ways a guide RNA for this telomerase protein.